Hello, everyone. My name is Efi Melista, and I am happy to welcome you at Omixon's user group meeting at the 2020 ASHI virtual conference. I would also like to take the opportunity to thank the ASHI organizing committee for making this conference a reality and also for making it available to everyone worldwide, especially during these difficult times that we are living today. Our user group meeting today will be focusing on advances in HLA typing and transplantation and OmniType Perspective and sugar-sprinkled transplants, ABO in transplantation. I will begin with a short corporate overview and talk a little bit about the company for the new people in the audience. I will continue with some new product announcements that I am very happy and very excited to share with you today. And then I will continue with introducing our guest speakers that will be talking on two separate topics. Of course, we will be finishing off with a Q&A session. So feel free to keep your questions to the end or to put them into the questions and answers box on your platform. So to begin with, Omixon is a global transplant diagnostics company with 57 people from 7 different countries, 48 people located in Hungary, 4 people in Europe outside of Hungary, and 5 people in the Americas. We also have R&D facilities in Budapest, both for assay and software products, but we also have several R&D partners located, but not limited to, Pennsylvania, Iowa, and Germany. We currently have 3 contracted manufacturing sites, both in USA and in Germany. And in order to better support our customers worldwide, and especially to make sure that we have plenty of redundancy to handle logistics challenges, we have 4 different warehouses in the USA and also in Europe. But as I mentioned, Omixon is, although Omixon has several employees located worldwide, we also do have a very wide distribution network to better support all of the HLA laboratories and beyond worldwide. So as you can see from this map, we pretty much cover the entire globe. So we are a global molecular diagnostics company right now with headquarters in Budapest, but also with offices in Cambridge, Massachusetts. Our existing products include Holotype HLA, which is an HLA typing assay and software product, Monotype HLA that is used for disease associations, Monotype ABO that is used for molecular blood group typing, and Monotype MIC-A, as well as HLA Twin that is a software designed for clinicians, and HLA Explore that is a software designed for researchers. We also have 2 new products, one of which we will be talking about today in more extensively. We have OmniType, which is the multiplex HLA typing assay and software product. And we also have the Azure Seq 200 CE, that is a SARS-CoV-2 detection test using qPCR. So let's move into the new product announcement, which is the more exciting part of this presentation. So first of all, I would like to introduce OmniType. So OmniType is Omixon's new multiplex genotyping product for the comprehensive gene amplification of multiple HLA loci and sequencing on Illumina platforms. HLA Twin, that was the only software that was specifically designed for HLA data analysis by NGS, is included in the kit. The combination of the 2 are now offering fast and accurate genotyping for everyone. So what do we know about the OmniType? So OmniType went into an early access program that was launched in January of 2020. The assay has been under active EAP for about 5 months, and we did have a couple of small setbacks due to the COVID pandemic. However, they actually worked in our favor more than anything else at this point. It underwent 2 iterations of improvements based on feedback. So we had phase 1 testing, where 1 round of iterations took place in order to lead into phase 2 testing with some more iterations that took place and then led us to the OmniType version 1.0, which is the final product. We had 16 participants worldwide under this early access program who performed more than 30 sequencing runs and tested more than 500 clinical samples, both from blood and buccal swabs. So let me continue with a little bit of an OmniType workflow overview. So some of the advantages and some of the benefits of this particular product is that it is able to offer DNA to library in about 6 hours. The hands-on time is approximately an hour and a half or 90 minutes. All 11 loci are amplified in a single tube. So essentially you just need one amplification tube per sample that you're processing. It does have a very fast library preparation workflow, and it does contain pre-mixed and pre-aliquoted library preparation reagents in order to reduce the risk for use errors and make hands-on time and pipetting steps as little as possible. So with all of this, you essentially get sequencing results early in the second day. The second product that I would like to present to you today is the Azure-Seq 200. This is a CE-IVD SARS-CoV-2 detection kit that is available currently in EU only through Omixon. It does use a real-time, a reverse transcriptase qPCR-based technology. And the biggest advantage that it has is that it offers direct processing with no need for viral RNA extraction before the test. The results can be obtained in approximately 120 minutes from beginning to end, which makes it very, very fast. And it is also very highly sensitive with the ability to detect 0.5 copies per microliter of sample. And with that, I would like to introduce our first guest speaker for today, Maja Nørgaard, who is a biomedical laboratory scientist at the tissue typing laboratory at Aarhus University Hospital. Maja has been working with HLA typing for 5 years, and Maja and her team were one of the participants in the early access program for OmniType. So today she will be sharing with us her experience and her OmniType perspective.
Thank you for the introduction, Effie. And hello, everybody. My name is Maja Nørgaard, and I'm a biomedical laboratory scientist here at the Tissue Typing Laboratory at Aarhus University Hospital in Denmark. I will be talking about Omixon's newest product, OmniType, a new kit for high-resolution HLA typing with NGS. My agenda today is a short introduction to our current workflow with holotype HLA, and then I will continue with OmniType, the workflow, our test in the laboratory, quality, results, and a conclusion in the end. I want to shortly go over the holotype HLA workflow. Most of you might already be familiar with this typing kit. We have been using Omixon's holotype kit for routine HLA typing in 4 years now. And we use both the 2411 and the 24/7 kit, depending on how many loci must be typed. Most weeks, we analyze 24 samples on this fully automated system, Biomec 4000 robot. One pipetting robot for the pre-PCR and one for post-PCR. In our laboratory, we type both donors and patients for stem cell transplantation and solid organ transplantation with next generation sequencing. We use 2 different techniques, one for the initial HLA typing and one for the confirmatory typing. We do this because all techniques have different advantages and disadvantages. And when we collect 24 samples for one NGS library, the costs are actually lower than when typing with a low resolution HLA PCR kit. Day 1 starts with sample preparation, application. Day 2, we do the quantitation and library preparation. On the 3rd day, we do the site selection of the library and sequencing on the MySeq. The last day, the 4th day, we use for data analysis in HLA TWIM software. This automated system gives us less hands-on time that we can use elsewhere in the laboratory. There is traceability throughout the whole process, from amplification to the finished library in the end. Also, this automated system gives us reproducible results. Very important factors to take into account when you are choosing and implementing a new technique or method in the laboratory. Here is a slide workflow of the time needed for this OmniType has been reduced to only 2 days. Within 1 day, you are capable of doing the DNA extraction, amplification with PCR of 11 loci in one multiplex PCR reaction. These amplicons are quantitated with the qubit, followed by a normalization with water. Library preparation consists of fragmentation, adaptive ligation, and indexing PCR. The pooled library is size selected with beads and quantitated with qubit before sequencing on the MiSeq. The second day is for data analysis in HLA twin. We were very happy to participate in OmniType's early access program for OmniType. We have done some testing of OmniType here in our laboratory. We tested 1 × 24 samples in phase 1, and we typed 2 × 24 samples in phase 2. And the final version of OmniType for research use only just got released, and we of course would like to try that one as well. For the testing with OmniType, we use controls from external quality programs and patient and donors from routine HLA typing here in the laboratory. I found the instruction for OmniType very clear, precise, and straightforward, ready to use, so we just went on with the typing here in the laboratory. This new workflow with OmniType has a lot of remarkable advantages. The multiplex PCR significantly reduces the risk of exchange between reagents and pivoting errors when mixing or creating the master mix. When using the holotype kit, you have to prepare between 7 and 10 master mixes. With OmniType, you only have to do 1 master mix with this multiplex PCR. The kit comes with pre-mixed reagents for library preparation, and this again reduces the risk of pivoting errors and increases the opportunity to have these reproducible results. When this new NGS method is implemented in the laboratories, we do not need any new equipment or instruments. In fact, we need less instruments and reagents. For example, these plastic PCR plates and quantitation instruments like fragment analyzer and reagents for this quantitation. This also makes this method cheaper. OmniType has a reduced time. Currently, we are using 4 days with the holotype HLA, but with OmniType, we can get the results within 2 days. We, though, have a huge limitation here at our laboratory, since we do not have our own MiSeq. All MiSeqs are gathered at a core facility here at the hospital, and because of that, we will need 3 days for our library preparation and sequencing. This reduced time makes it more flexible for us. We can collect enough samples for one run, and to look at it. It's more flexible. The last point out here is due to process. As mentioned before, find this hands-off time, traceability, reproducibility, really important. When we did one of the tests, we accidentally chose a wrong program on the thermocycler. This and mix up within the steps can be avoided when the process is fully automated. Even though we have good instructions in the laboratory, mistakes happen when a method is done manually. We all know that. OmniType has a couple of disadvantages. When talking about the automation, the reagent volumes to pipette have a significant importance. For the library preparation, reagent volumes between 1 and 3 microliters must be pipetted, and that is quite a small amount for a robot to handle and can lead to pipetting errors. Also, sufficient dead volume must be present because the pipetting robot has a higher waste. So a higher reagent volume will potentially increase a successful automated process. This multiplex PCR could potentially have some limitations like dropouts. These dropouts might be specific alleles or loci. And this is always a concern when going from singleplex to multiplex PCR. And I would like to test this kit further to fully exclude this possibility. Let's take a look at some of our results. All 3 libraries have perfect concentration. However, we experienced some challenges with the sequencing on the MySic. Unfortunately, we used a cartridge with bad chemistry, and 2 sequencings with the OmniType phase 2 had densities below acceptable. They were both around 450. The passing criteria is between 500 and 1200. Together with the Omexon team, we conducted troubleshooting regarding this issue. And along with another laboratory, we saw this low density when sequencing on MySic. And this troubleshooting affected a change in the protocol when featuring the library before sequencing on the MySic. We repeated the sequencing with the same library and got good density and yield. However, this time the cluster passing filter was 68%, and the passing criteria for this is it must be above 80%. I have no doubt that we were struck by bad luck, but I would really like to see some good sequencings with all criteria passing quality control here in our own laboratory. Now let's move on to some real HLA results. I am only including samples from 2 runs, the 2 runs from phase 2. The results are from sequencings at Omexon when we conducted the troubleshooting. In those 2 runs, there were 48 samples according to 96 alleles from each loci. And this list, this table lists how many concordant alleles were found, also in percentage. All over, the results look really good, and the HLA types analyzed with OmniType are the same as the reference HLA types from Holotype. 7 loci have 100% concordance, but let's take a look at some of the loci with missing concordance. HLA B, C, EQ, B1 have a total of 5 discordant alleles. 4 of these are dropouts, meaning the alleles are not found or called as best maps due to incorrect phasing and cross-mapping in the software. 1 allele had a dropout because of low amplification. HLA ERB1 has a total of 8 discordant alleles. All 8 alleles are within the allele group ERB104, and they are actually not dropouts, but due to extreme allele imbalance, the software calls rare alleles or novel alleles. A heterozygous HLA type is found, but it's not impossible to determine the HLA type. ERB104 has this well-known infant 2, which is significantly longer than other ERB1 allele groups. This is a well-known problem or issue when designing and creating the amplification of ERB1. This multiplex PCR also has difficulties managing this issue, and Omnicom's team must find a solution to this. We need to be able to type ERB104. This was some of the challenges with OmniType, but the results also showed some advantages. Omnicom has a list describing known product limitations, and dropout before is one of them. And this is an example of a dropout in holotype HLA with the software. HLA Twin gives a warning message. And due to linkage between ERB1 and ERB4, we would expect this ERB4 type to be shown. Reading this sample with a new extraction of DNA and a new amplification makes the same result, this dropout. The same sample was analyzed with OmniType, and it gives a perfect result, ERB4 0101. Another example is this homozygous ERB4 0103. We found this with holotype. It is only different at 4th position, which has no clinical relevance. Notice that there is no warnings or failed quality parameters. When typing this sample with OmniType, the result is a clear heterozygous ERB4 0101 0103. The same sample has been used in both methods. This is an example of a novel HLA-B allele. The allele has not been reported yet, and therefore has not got an official name in the IMDT database. And when we are having a look at this alignment, it shows that there is a single nucleotide polymorphism and a change in the amino acid in the coding region exon 2. This allele was first detected with holotype, but also OmniType shows great coverage. In fact, more than 100 reads support this novel position. This was a short review of some of our results from the testing with OmniType here in our laboratory. And I just want to summarize what I have been talking about. After testing this new OmniType kit, proves that it is an easy to use and streamlined process for next generation sequencing. This fast turnaround time makes it attractive to use for routine HLA analysis in the laboratories. This process seems straightforward to implement, and no extra instruments and reagents are necessary to buy if you're already doing next generation sequencing. With a couple of small adjustments, this method could be automated. The results shows promising HLA typing, but more testing and development must be done. Especially the software needs some adjustments, and the multiplex amplification needs improvements, and we have to avoid these dropout of specific loci and alleles. But when these adjustments and improvements have been done, I see a great potential potential in Omixon's newest kit, OmniType. And I really look forward to work even more with this kit. So thank you very much for listening to me. If you have any questions, I'm sure you may contact Omixon at this email and they will help you. I hope you will enjoy the rest of this virtual conference at ASHI. Bye.
Thank you, Maja, for your presentation. That was great. For our next guest speaker is going to be Halpin, who is a laboratory scientist in the Clinical Histocompatibility Laboratory at the University of Alberta Hospital. Anne is also a PhD student in the laboratory of Dr. Laurie West, also at the University of Alberta, where she is focused on immune risk assessment in pediatric heart transplantation, including the detection of ABO antibodies to assess risk in ABO-incompatible transplantation. So today, she's actually going to be talking to us about something slightly different in the HLA community, and the title of her talk is Sugar-Sprinkled Transplants, ABO in Transplantation.
Thank you so much to Omixon for inviting me to talk about this topic today. ABO has become an area that's very near and dear to my heart. So I hope that over the course of the next few minutes, I inspire a little bit of a spark of passion of ABO in you, if that didn't already exist. And we can talk about the complexities of ABO and how it relates to transplantation, and particularly how an HLA person might want to think about ABO. Let me just go to my next slide here. I'm going to start by introducing the team of people that I'm very fortunate to work with on this project. So Dr. Laurie West has a long, rich history in ABO-incompatible transplantation and research, and she has many amazing people in her team. So Tess and Jean are the technicians who do all of the heavy lifting on these projects, as well as Bruce, who is always constantly herding the cats on this team, and all the other people on this slide who've contributed in many ways to this work. So this quote I like to think about as a starting point for talking about ABO blood groups, because we think about ABO blood groups as very simple, and I certainly did when I first started thinking about ABO. I mean, I'm an HLA person. I like very complicated things. I want lots of complexity and polymorphisms, and ABO blood groups just didn't seem like they were that exciting to me. However, I was very wrong, and I think that over the next few minutes, hopefully you'll agree with me that there is a lot to think about when it comes to ABO, and that even though it's a simple beginning of 4 phenotypes, there's a lot more to consider, particularly when it comes to transplantation. So as I said, there are 4 major blood groups. I think that's probably not news to anyone listening to me right now. Blood groups A, B, AB, and O are the 4 main phenotypes that we think about when we think about ABO blood group antigens. However, these aren't really just blood group antigens. They're really a histo blood group system, because the ABO blood group sugars, which I'll describe the sugar part of that in a minute, they are not just on our erythrocytes, but they are sprinkled all over our whole entire body, including our organs, which is why we have to think about them in transplantation. And because these are carbohydrate antigens, the antigens themselves aren't directly encoded for by our genes. The genes actually encode for glucosal transferases, which then decorate our cells and tissues with these blood group sugars. And we also naturally form antibodies to ABO antigens that we do not have. So different, again, than HLA in that sense, where there's a non-self HLA exposure, which results in antibodies. ABO blood group antigens, we have antibodies to any blood group antigens we lack. So myself as an A person, I have anti-B. An O person has anti-A and anti-B, and a B person has just anti-A, and an AB person has no antibodies to blood group antigens, theoretically. So when we think about ABO and solid organ transplantation, it is one of the very first things that we consider when we're listing a patient. We think, what's their blood group? And then their donor selection pool is immediately tailored to that. And so we have ABO identical donors, as I've shown here, if, for example, an O to an O, or we may have an ABO compatible donor in, like, for example, an O to an A person. But also you can have ABO incompatible transplantation, where, for example, you would transplant an A heart or kidney into an O recipient. The ABO incompatible light, as some people like to call it, is more where we have blood groups like an A2, which is a slightly less A sugar-coated organ, into an O person. And I'll talk briefly about blood groups such as A2 in a few minutes. And again, these preformed antibodies to ABO blood groups are why we think first and foremost about ABO when we're considering a donor pool for a recipient, because these antibodies, if we choose to ignore them, would be very dangerous. They would result in hyperacute rejection. So blood groups. There are many subgroups to ABO, and often that's what we think about when we're thinking about some diversity in the system. So the most common example of that is that you can be an ABO A1, like me, or you can be an ABO, what we call A2, which is a different gene encoding for the glucosal transferase that is decorating our cells and tissues with ABO-A, and it's less efficient. So you end up with less antigen and slightly different forms of that antigen, actually, on your erythrocytes as well as your organs. But when we're testing for this in the lab, typically transfusion medicine labs are using what we call an A1 lectin, which is kind of like an antibody. So we're not actually saying it's an A2 specifically, we're saying that it's not an A1. And that's an important difference because there's a lot more diversity than just A1 versus A2. So ABO blood groups don't have the diversity of HLA. Nothing really does, but there is diversity within the system at a genetic level that we can differentiate. And we know there to be 200 ABO alleles. Maybe there's more, but there are 200 that we know of. And this is just a screenshot from the ISBT website where they list a number of different alleles. Now, these alleles here are shown from chromosome 9, which is where you find the ABO A and B glucosal transferases. But it's also important to consider that on chromosome 19, there are fucosal transferases that encode for our H antigen, which is ABO-O, and the secretor status, whether or not we secrete ABO into our tissues. So genotyping, there are commercially available ABO genotyping kits, and these primarily focus on chromosome 9, and I'm not aware of one that looks at fucosal transferases as well, but the ABO genotyping kits focusing on chromosome 9 is probably the most relevant in transplantation. And these then provide information about the glucosal transferases that we happen to carry in our DNA. And this, again, is telling us about those transferases. We still don't exactly know how our cells and tissues are decorated. So using things like immunohistochemistry to look at tissues, or using flow cytometry studies with red cells would give us a little bit more information about that sort of endpoint. How did that decoration occur? What does it look like? And you can also use serology testing to do that, which I'll talk about in a minute. So this association between our genotype, the resulting antigen, and the antibodies that we naturally make isn't fully understood. And I think we have a lot of work left to do there to figure that out. So when it comes to the blood group antigens themselves, there's diversity in how these sugar structures look. So we know that there are 18 different sugar structures, I guess I'll call them, that could be potentially present, and at least 4 of them we know to be present in humans. So this, me as an A1 person, on my red cells, or erythrocytes, I have the antigens 2, 3, and 4, the subtype antigens. This is not the same as the A2 group. These are the subtypes of the sugar structures themselves. These are on my erythrocytes, but if you were looking at my, let's say, cardiac vascular endothelium or any of my endothelial surfaces, you would only see the subtype 2 structure. So that's really important to consider if you're doing an antibody assay using red cells as your surrogate for what might be relevant for the transplant, because perhaps you're detecting something that isn't on endothelium. So another interesting difference between ABO and HLA is IgM. So because this is a carbohydrate antigen and our immune systems respond differently to carbohydrate antigens than they do to proteins, we cannot ignore IgM, and we have to consider this in our antibody investigations for ABO. So the IgG antibodies are the focus of protein antigen investigations such as HLA, but with ABO, we look at both. And these are what we call T-cell independent responses to carbohydrate antigens, although I think that dogma is potentially a little outdated and we need to think a little differently about that with regard to ABO, but definitely IgM is very relevant. And also it's important if you're using strategies such as plasmapheresis to remove antibody, because IgM antibodies tend to live more in our intravascular spaces, so pheresis might more readily remove IgM than IgG. So if you're using a method to detect an antibody that's specifically looking at IgM only, like agglutination tends to do, we'll talk about that in a minute, you may be underestimating IgG burden. So how do we look at ABO antibodies right now? Hemagglutination is a very robust method that's used still to this day in transfusion medicine labs. And Dr. Karl Landsteiner was the, I guess, the father of transfusion medicine. And in 1901, he showed us that there are these 4 blood group antigens, and he did that by using red cell agglutination. And the whole field of transfusion medicine was really born as a result of his work. And so clinical transfusion medicine labs today still use this methodology to perform ABO blood group typing with using antisera to A and B, and also to measure antibodies using erythrocytes as targets and measuring how much antibody is in the patient using agglutination as a final output. So when it comes to hemagglutination, there's quite a bit of diversity in how you might run that test. You can use different temperatures, such as room temperature or 37 degrees. You may or may not have different incubation times. You might use a gel card system, which is something a lot of transfusion labs now use, or tubes. You might be using commercial erythrocytes or panels from within your own lab. You may or may not use DTT. You may or may not use anti-human globulin. Any of you who've worked in HLA for a while are probably feeling flashbacks to our serology days right now, because this is, you could use this almost exact same slide with the exception of the gel card system to describe complement-dependent cytotoxicity variation in testing. And just like in HLA antibody testing by CDC, when you use hemagglutination, you see a lot of diversity in responses from labs who are testing the same sample. So this slide is just an example from a College of American Pathologists proficiency testing exchange. So you can look at the pink hashtag curve here. These are room temperature room temperature tube detection of A1 antibodies, or I guess in this case, they probably used an A1 cell though in most of these labs. And you can see that there were titers from 1 in 16 to 1 in 512 reported. And if you used anti-human globulin and room at 37 degrees, the curve shifts a little to the right, but there's still a lot of variation in what labs are reporting and seeing. So how can we improve ABO antibody detection? Well, HLA people have turned to Luminex in the past, and I think this is an excellent opportunity for us to look to Luminex for ABO blood group antigen antibody detection as well. And so our group has developed a Luminex bead-based single antigen assay to define and detect ABO antibody patterns. And this, some of this work was done by one of our students, Janet Zhao, an undergraduate student who worked with us. And you can use this bead coupling protocol, and you can couple antigens to beads using methods like this. The important thing, though, is what antigens are you using? And for us, we were very fortunate to work with the carbohydrate chemists who are here at the University of Alberta, who have manufactured these antigens for us, and they're bovine serum albumin coupled so that we can use a protein coupling procedure. And these 3 papers highlight their methods for creating these antigens for us. So once you've coupled them, you want to make sure that they're actually present. And this slide shows us here how so the blue bars are the monoclonal antibodies to A, the yellow bars are monoclonal antibodies to B. And you can see that there's really strong, clear antigens present on all of these individual beads, the trisaccharide, which is the terminal sugar, as well as the structures Roman numeral 1 through 6 on the beads for us. So what does this look like then when we start testing people? We did another study with another fantastic undergraduate student shown here, Morgan Sosniak. And so we looked initially into 28 people. We've looked at a much larger group now. And we looked to see, well, what antibodies do we see in normal healthy adults? And we compared this to hemagglutination. And what we found is that there are some really interesting differences in the antibodies that you can detect even within the same titer. So if you look at, I'm not sure, I don't know if I have a bar here, or an arrow, but if you look at the A titers on the left side of the screen, you can see there's a 1 in 16 titer, and the blue dots are the IgG MFIs, and the burgundy squares are the IgM MFIs. And so for that very same titer of standard hemagglutination, there is a really wide variation of the amount of IgG and IgM antibody present. And this is true throughout this testing. And so if we're using IgM as a surrogate for how much IgG antibody might be present, we're going to be in for some surprises, because focusing on here the A2 antigen specifically, antibody to this in both healthy controls as well as a pediatric bone marrow transplant group that we studied, you can see that the amount of IgM antibody doesn't necessarily reflect the amount of IgG antibody for any individuals. There's a few who have similar MFIs to both, and you can't compare IgG and IgM apples to oranges, or apples to apples. Here they are, apples to oranges. You can see the patterns are all over the place. Some people have very little IgM and more IgG, and vice versa. So thinking about both of these isotypes is important. Also important to consider is that some people, even if you're blood group O or B, all of whom we would expect to detect anti-A, that they can have varying amounts of antibody. And so this is perhaps representing some opportunities in ABO-incompatible transplantation for us. So also another thing to consider is the reproducibility of this assay. So as I showed you with the CAP proficiency testing survey data, there's a lot of variation, as you might expect with a method that's serologic and has variation from lab to lab. So we wanted to see how would these beads perform if we sent them out to many different labs. So I talk about this more in ASHI poster 7, just plugging the poster there. But there are, this exchange, we had 9 labs participating with us, and we sent the same 10 serum samples, the same reagents, and an SOP and Luminex templates out to everybody, and then we had everyone test these samples. And we found that there was really great reproducibility between these labs, and so this is an example of an IgG ABO antibody to our positive control sample, just as a representative example of this. But you can see that there's quite good comparison between labs. Some labs used a 3D instrument, some used a 200, so that did change the MFI outputs a little bit, particularly in the high MFI samples, but overall, really nice concordance between these labs. So in summary, ABO A and B subtype antigen expression, or decoration as I like to say, it differs between red cells and tissues. So using red cells as a surrogate for antibody detection it comes with some lack of specificity that we may want to consider when it comes to solid organ transplantation. ABO genotyping tools exist, and these can be used to better understand the complexity of the histoblood group system, particularly if we're combining that with antibody detection or immunohistochemistry, but just even in general, I think that we need to think a little bit more than beyond our 4 phenotypes that we see. So the Luminex bead tools that we already use widely in our HLA laboratories, that same platform can be used to detect ABO blood group antibodies. And so that's a nice opportunity for us to use this technology and the equipment and the expertise we already have to think about detection of ABO blood group antibodies. I do think we need to do this in collaboration with our transfusion medicine colleagues, because they of course have expertise in this area and that we should take advantage of. But I think that the tools that we bring to the table and the expertise would make for great collaborations. So better characterizing ABO antibodies allows us to really understand and measure the risk assessment in ABO-incompatible transplantation in a way that we currently struggle to do using erythrocytes as a surrogate for the tissues that we're transplanting. And I talk a little bit more about this in the new technologies oral abstract session, abstract number 135. So again, just plugging another piece of our science. And I think, and I really want to stress this, I think that ABO-incompatible transplantation, while may not suit every single recipient, may be something that is a good opportunity for our highly sensitized patients for whom we have very limited donor pools, or for the living related donor who has an HLA identical sibling but who is the wrong blood group for them, We already do this widely in ABO-incompatible pediatric heart transplantation, which is a little different because those infants have not yet made their own blood group antibodies. But that whole premise was born out of the necessity that that group of patients has very high waitlist mortality. So perhaps thinking about some of our most challenging patients and whether or not ABO-incompatible transplantation is a good tool that we could put in our toolbox to deal with that and to provide them more opportunities is something that we could talk a little bit more about, and particularly if we were able to use tools like genotyping and ABO antibody detection using single engine beads to go along with those types of transplants. So we have many people to thank here. Dr. Lori West, of course, and the team that I already described to you earlier in this presentation. Dr. Chris Cairo and Dr. Todd Lowry are our chemists extraordinaire who make these antigens for us. The HLA lab staff who let me go over with all my little ABO beads all the time. And of course, that includes Trish Campbell and Esme Dyke in our labs here. And Dr. Simon Urschel, who is a pediatric cardiologist here, who really helps us with patient recruitment and providing excellent clinical feedback on this work. And of course, all the patients and volunteers who provide all these samples, consent to be part of our studies. We couldn't do it without them, and all of our funding agencies. So thank you very much for listening to me, and I hope that if you have any questions that I'm able to answer them for you.